to the heart, or so that's what we're talking about. Synapse is a synaptic transmission. What is a synapse? Just a specialized point of contact between a neuron and another neuron, or a muscle cell, or possibly a glandular cell. It's how information is transmitted from one neuron to another, from a presynaptic terminal to a postsynaptic terminal or membrane. We have two major types of synapses. The first is an electrical synapse, and we'll just briefly talk about that. It uses what's called gap junctions, which are just these junctions that are directly connecting two cells together. You tend to see these more between muscle cells and glandular cells. Some of like your glial cells also have these gap junctions, like the astrocytes and so forth have some of these gap junctions. There's a pre and postsynaptic membrane, and they're really close. They get even closer than a typical synaptic membrane, so they get really close together so that these channels can cross between both of those membranes. Okay? Typically use a type of a channel called a connexin, or a protein called a connexin. That's these channel proteins that allow for that direct communication. They're big pores, they're fast. So especially when you're trying to coordinate the activity, say the heart muscle, so all these cells, can contract at almost the same time, and so you get a spread of the electrical signals that can passively move through these gap junctions, or calcium can move between them in muscle cells, and so you get these rapid contractions of lots of cells at the same time. Or for glands, if you want to have a gland that's activated so it releases whatever hormone or substance that it's releasing all kind of at the same time. So cardiac, smooth muscle, and glial cells. Just a real kind of a close-up picture here showing, the connexins right here. These proteins that are passing through both membranes, and in this case, they get really close together here. This 3 nanometer kind of distance, 3.5 nanometer distance there, so you can have that direct connection. So that's electrical synapses. Now let's talk about chemical synapses, which are by far the most common way we transmit information or communicate between neurons and other neurons, or neurons and muscles and neurons and glands. You have a presynaptic terminal, which is that end of the axon, right? Also sometimes called a axon terminal or a terminal bouton. Those are all synonymous terms. So presynaptic terminal, axon terminal, or terminal bouton are all the same thing. Then you have a cleft, or a space, between that neuron and the other neuron, or that neuron in the muscle cell or neuron in the gland. And then on the next neuron or muscle or gland cell, you have a postsynaptic terminal. Okay, it's a specialized area of a target neuron or a target cell. And that postsynaptic terminal has typically receptors in it that are specific to the neurotransmitters that are being supplied by the cell nearby or the neuron nearby. Then you have a neurotransmitter. Okay, these are chemicals that just contained in small vesicles that stimulate or activate the protein channels on that postsynaptic membrane. You can have connections, when we're talking about neurons specifically, so we're just talking neurons here, you can have what's called an axosomatic connection, an axodendritic connection, or an axoaxonic connection. I'll show you a picture here of that in a second. So here's our terminal bouton, presynaptic terminal, axon terminal, right there, that little bulbous area at the tip there of the axon, or a branch of the axon, okay? And you have our little vesicles here that are within that are containing the neurotransmitter, and here's our postsynaptic membrane and postsynaptic terminal with some specialized receptor sites. Okay? I think I fixed it. I wasn't going to say anything, but I think I fixed it. So anyway, will not be defeated. And our synaptic cleft right here, synaptic cleft, which is, I don't know what the exact how many nanometers that is, if it's 10 nanometers, whatever. It's more than 3.5 nanometers. Here's the different ways that one neuron can interact with another neuron. So you can have this would be an axon terminal that's directly communicating with the soma, or the cell body. So that's an axosomatic connection. Or you can have, this is probably the most common right here, is there's a better chance, because you have lots of dendrites, there's a better chance you're going to have an axodendritic synapse because there's just a lot more dendrites with the little dendritic spines, lots of surface area to end up bumping into another neuron's presynaptic terminal or terminal bouton. So axodendritic are probably by far the most common interaction you're going to see between two neurons. But then we also can have a interaction down here. It's called an axo-axonic. So right there at that terminal bouton, that presynaptic terminal, you can have another presynaptic terminal that directly influences this other presynaptic terminal. And we'll talk about the concepts of presynaptic facilitation and inhibition. Okay. So here's a summary of events that happen at a chemical synapse. First of all, you have an action potential that is being sent down that axon, right? And it finally reaches that presynaptic terminal, the end of that. That action potential, when it gets down to the presynaptic terminal, there's a bunch of calcium, voltage-gated calcium channels down there, right? Remember up in the axon hillock near the soma where the action potential started? There's a bunch of voltage-gated sodium channels up there. Down the axon terminal, there's a bunch of voltage-gated calcium channels. Okay? So once the action potential gets down to that presynaptic terminal, it opens those voltage-gated calcium channels. That causes calcium to come in, and the calcium moving in, well, I don't know exactly how or why, but that triggers the vesicles to move to the presynaptic membrane. Okay? And once it connects or touches the membrane opens in a process called exocytosis to allow neurotransmitter to be released from that presynaptic terminal into the synaptic cleft. Okay? And that neurotransmitter crosses that synaptic cleft because it's a close enough distance. Most of the neurotransmitter, not all, it will end up binding to some of those receptors that are open right now that it can bind to on that postsynaptic membrane, in the postsynaptic membrane receptor. The postsynaptic membrane channels then change shape. Okay, so they're binding to ligand-gated receptors. So the protein channels on that postsynaptic membrane are called ligand-gated protein channels. Okay. They cause the protein channels to make a change. It could open more ligand-gated sodium channels, which would then help to depolarize the next cell. Or that particular neurotransmitter may be more specific to chloride channels, which opens those and causes negative ions, chloride ions, to come into the cell and actually inhibits the next cell. Okay, so you can actually get this. You can either get excitation or inhibition depending on what is happening with the nervous system and what function we're talking about here. Okay. So it can either cause a direct change in that, or if it binds to what's called more of a protein that's associated with this G protein pathway, kind of longer acting changes to the cell. Okay. Neurotransmitter stays bound for just a brief period of time, and then it's released. Then it's either broken down by enzymes in that area in the synaptic cleft, or some of those neurotransmitters are have some reuptake by the presynaptic terminal. Okay. And so it recycles it and through the process called endocytosis, so it sucks some of that back in, or the leftover pieces of those neurotransmitters, so it can be taken back up to the soma and recycled and used again. Or you have some like astrocytes or glial cells nearby that will kind of suck up any leftover neurotransmitter that's also floating around near that synaptic cleft that isn't no longer needed and wants to shut down that process of communication, because most of the communication is temporary. It's pretty short acting. Okay? So here is the process in pictures. Action potential coming down. You see our flip-flop of charges here as it goes down, and most of the time it's jumping between those nodes of Ranvier through the voltage-gated changes in the voltage-gated sodium channels. But once it gets down to the end here, and you start to see this gets positive here at the presynaptic terminal, that opens some voltage-gated calcium channels down here at the end, and that allows calcium to come in, because you actually do have a calcium pump. Like we have a sodium-potassium pump, but you also have a calcium pump that pumps calcium out, so calcium always is a higher concentration on the outside, so it wants to come into the cell, okay? Even though there's a positive charge down there, even though it's positively charged inside, there's enough of a concentration gradient to bring some calcium into the cell there. That triggers those little synaptic vesicles to move to the presynaptic membrane and then release their neurotransmitters. So there they are moving towards that. Then here's the exocytosis, and you're dumping the neurotransmitters into the synaptic cleft. And then we get binding of these neurotransmitters into specific receptors that are specific to that neurotransmitter, okay? Or specific to things that very closely mimic that neurotransmitter, okay? So when we talk about drugs, sometimes we have drugs that are trying to mimic a neurotransmitter and bind to those receptor sites that normally be bound by a neurotransmitter. We can get drugs that are close enough to look like it that they'll hang out in that receptor. Maybe not always as long and not as well, but it's pretty close enough that it can get some activity there, okay? And then eventually, these vesicles are recycled here. This causes the activity. In this case, this happened to fall on ligand-gated sodium channels, which allowed positive charges here. So here we're getting an excitatory postsynaptic potential, which may or may not lead to action potential in that next neuron. We don't know unless we have enough summation, you know, either temporal or spatial summation of other neurons nearby that lead to that enough positive charges coming in to lead to action potential in the next neuron. And then some of that they don't show this process, but neurotransmitter, they should have showed some of these reuptaking some of that neurotransmitter, and then they are through retrograde transport on the microtubules, these little synaptic vesicles, and some of them have some neurotransmitter they've grabbed back up, will go back up to be recycled, repackaged, and then there's always more coming down this way through anterograde transport down the axon to replace. Okay, so there's constant conveyor belt kind of action going on. So let's talk about electrical local potentials at synapses, which is, we've already mentioned this multiple times, but we can either have excitatory postsynaptic potentials, like this one's an excitatory postsynaptic potential because it actually caused depolarization. Positive charges came in because it bound to a ligand-gated sodium channel in this case. So that's an excitatory postsynaptic potential that causes depolarization that may lead to an action potential with summation. We get that threshold, minus 55 millivolt. Or we can have inhibitory postsynaptic potentials. It's called an IPSP. This causes hyperpolarization of the next neuron or cell. Okay, makes it less likely to be active or have an action potential or to contract or whatever. That's an inhibitory postsynaptic potential. And that's typically because you have a receptor or a neurotransmitter that binds to a protein channel that allows chloride to come into the cell. Chloride's a negative ion. So you also have some chloride pumps that keep chloride on the outside of your cell, and chloride will come into the cell and cause it to get a little more negative, and so it's less likely to create an action potential, or the gland is less likely to contract, or the muscle is less likely to contract because it's been negatively, you know, and it's been inhibited through its negative. Or it can also, another way to do that is open up some more potassium channels. Even though you have those leaky potassium channels, there are some you can open up some potassium channels to let even more leak out, and so that even makes a little bit more negative that resting membrane potential. Okay. These two processes basically regulate the excitability of your neurons, okay, in the nervous system by either excitatory or inhibitory. You always have to have a way to turn things up or turn things down. And we talk about most organ systems, whether it be your heart, lungs, brain, kidneys, liver, you got to have ways to ramp up function and ramp down function. But usually your set point's usually somewhere in the middle, you know, whether it be muscle tone, whether it be our nervous system, we're somewhere in the middle, and we go up from there or we go down from there through excitation or inhibition. We always have these balancing systems in every system in our body. So this is one way we do that. So you can excite it or you can inhibit it. depending on the need at the time. Okay. So this is just showing you, again, in some pictures here. Here's our resting membrane potential here on the left. Neurotransmitter binds to a ligand-gated sodium channel. A few sodium ions move in, and we go from our resting minus 70 millivolts. We bump up, but we don't get to threshold yet. But this is, maybe we're at minus 60 here. Okay, not to minus 55 yet, but that's an excitatory postsynaptic potential. We depolarize a little bit. Okay, got a little more positive on the inside because we let a few positive sodium ions in. Then here's the flip side. Here's some neurotransmitter that happens to be specific to some chloride channels. Okay, so it opens up some chloride channels, and now we allow a little bit some extra negative charges to flow into an already negatively charged inside environment. So we were at minus 70, now we're at minus 80. So even if I open up some sodium channels now, it's going to be harder to achieve my action potential because I have to go from minus 80 up to minus 55 instead of from minus 70 up to minus 55. Okay? That's an inhibitory postsynaptic potential. Let's talk about the concepts of presynaptic facilitation and presynaptic inhibition. This is basically when the amount of neurotransmitter that is released by one presynaptic terminal is influenced by the previous activity of an axo-axonic synapse. Okay? So here's what we're talking about. So presynaptic facilitation, this is this axo-axonic synapse. It's influencing this presynaptic terminal presynaptically before the synapse right here. Okay? And so what's going to happen here with presynaptic facilitation, this axo-axonic synapse may dump a few neurotransmitters into that area, which may open a few ligand-gated sodium channels. Also the ligand-gated calcium channels, but it may open and allow, it'll be a little more positive there when this action potential actually arrives. So the action potential is coming down. You basically primed it. You've kind of primed that presynaptic terminal, so that's already like getting excited, waiting for that action potential. When the action potential hits it, it's just going to dump even more neurotransmitter and create a stronger response than if you did not have this kind of presynaptic facilitation. If that wasn't there, you'd release X amount of neurotransmitter, but because you've already primed it and got it excited, it's going to release a little bit more neurotransmitter so that you're going to accentuate or enhance the amount of neurotransmitter then you're going to enhance the next activity with the next neuron. Now that could be, these neurotransmitters could be binding on inhibitory, like chloride channels that open up and make it more inhibitory, possibly, okay, that next neuron. But in this case, most times we're talking excitatory, okay? So prolongs that action potential, strengthens it. And then the presynaptic inhibition is the exact opposite. So in this case, we're probably binding to some chloride channels, making this presynaptic terminal a little more negative with a local potential right there as this action potential comes down. So the action potential gets there and you're inhibiting it a little bit. So when it does activate, you'll see there's less neurotransmitter, less synaptic vesicles that are going to be released there. So that's presynaptic inhibition. You get a little hyperpolarization down at the end when the action potential gets there. Yeah?
For the last slide, did you say it could also be working to reverse that or negative for the inhibitory effect?
I should probably not have gone there, but anyway. Yeah, no. So what it would do, so let's say you could have you could facilitate this release of neurotransmitter. Depending on the neurotransmitter, if this neurotransmitter binds to sodium, ligand-gated sodium channels, you're going to enhance this response and excite the next neuron. However, if you have presynaptic facilitation, if this happens to be releasing a neurotransmitter that's going to bind to, say, chloride channels here, I could actually cause this postsynaptic cell to become more inhibited, but through presynaptic facilitation. Does that make sense? So all we're doing is, with this interaction, is either facilitating or inhibiting how much neurotransmitter, how strong that response is. But those neurotransmitters themselves could either excite or inhibit the following cell. So we're not saying anything about the postsynaptically. We're talking about what's happening presynaptically. Does that make sense? Okay. So this is just another way that your nervous system has the ability to fine-tune and regulate all overall excitability, up or down. Okay, it just gives you another nuanced way to affect how the nervous system is regulated. And some of the things when we're training patients, if I'm working with patients with neuroplasticity, if I'm doing lots of repetitions of motor activity, I may strengthen some of those connections. That's one way I can affect neuroplasticity, is by strengthening connections or making those connections less strong, because if I'm not using certain neurons, then they don't have as many of these connections. And, and it can be both facilitation or inhibition, depending on, certain muscle groups I'm wanting to facilitate for a certain movement, but I'm also wanting to inhibit some other ones that aren't needed for that particular movement. So that's one way that we can influence, neuroplasticity, is through some of these complicated and diverse connections between the nervous system. And this is also how things like chronic pain and those kind of things could be influenced. So sometimes you get extra strengthening and you get too much presynaptic facilitation in a pathway that maintains pain, okay, when normally you wouldn't want it. So you have to then work on strategies to counteract that through movement therapy, through some of the modalities you're using, those kind of things. So, make sense? Synapses? All right. Let's talk briefly about receptors, the receptors that bind the neurotransmitters. We're going to just talk about two major types. We have synaptic receptors that are ligand-gated ion channels. The ligand is just a neurotransmitter or whatever molecule that's going to bind to that. Proteins that function like a gate. Okay, so these are these proteins that function like a gate. The gate opens when the neurotransmitter binds. There may be a slight delay. Some of them have different timing, but mostly it's pretty quick, though. It's going to happen pretty quickly with these ligand-gated protein channels. Once that thing binds, it's going to happen pretty quick. Okay, and then it's going to be pretty fast. It's going to open, but then that neurotransmitter is going to release, and it's going to close pretty fast. These are typically fast-acting. They just open and close as long as it's bound to the receptor site. This is the majority of synaptic activity is mediated this way through ligand-gated channels. So showing you, here's the neurotransmitter, also called the ligand. That's just a general term to describe any neurotransmitter or any substance. It doesn't have to be a neurotransmitter. Any molecule even is like the neurotransmitter, like medicines you might take, could bind to that. It's a ligand. It's acting as a ligand to bind to that receptor that's semi-specific. It's not 100% specific, but they're pretty specific to something that's similar in shape or affinity for it. And then that opens this channel briefly, and then as soon as that thing releases, it closes again, just about as soon as it happens. Okay? Then the other type of receptor, big classification of receptors, is called these G protein mediated receptors. Okay? So these are a little more complex. They tend to be slower acting. They generally are longer acting, and they're more modulators in the nervous system. So they typically are regulating larger networks of neurons, either raising excitability of lots of neurons that are kind of in certain regions of your nervous system and tuning them up or tuning them down. They don't affect the individual little, I guess they do in a sense, like connections between them, but they're regulating more overall static set points of certain regions within your nervous system that are associated with things like, could be memory or alertness or mood, all those kind of things are regulated by some of these G protein mediated receptors. And then some of our hormonal systems and glandular systems, and even some of we'll talk about the arachidonic acid system, which is partly related for heart contraction with the sympathetic nervous system. We'll talk about that. It's mediated by some G protein mediated receptors. Okay. It's also involved what's called the second messenger system, which we'll talk about. So here's kind of the simplest way that this G protein mediated system works here. So here's a receptor, and it's not a channel. It's a receptor. Okay, it's a protein that's embedded in the cell membrane, and it happens to have a complex of other proteins attached to it. Okay, and it's called the G protein. Okay, and the G protein is typically made up of 3 subcomponents: an alpha, beta, and gamma subcomponent. Not that critical to know all this stuff, but it's, just so you appreciate it a little bit here. In this case, what's happening here is a neurotransmitter, which really is a neuromodulator, because still called it neurotransmitter is the big classification. Then we have fast-acting neurotransmitters and neuromodulators, which are still all neurotransmitters. But so this is really become more of it's a neurotransmitter. It's more of a neuromodulator because it's going to modulate activity for longer periods of time. It binds to this receptor, this G protein-mediated receptor. That causes a change. It causes you to use. Some of this uses energy. Okay, so it converts GDP to GTP here. Again, not really that critical for you to know that. That process, though, of converting that causes, in this case, the alpha chain. So this is the alpha chain or alpha subunit to release. And it goes over and communicates with a really nearby protein channel. It may bind to that protein channel, and it may cause that protein channel to open, and it may last for several seconds, even to several minutes. It could, some of these. So these are versus milliseconds. So these kind of changes might take seconds to sometimes minutes, and so, but then once it's done its thing, it floats back over, and I don't know that it really floats. These things actually might even be closer together. They might almost be touching each other. That's not that critical. It's just that this is how the G protein mediated. This is one way G protein mediated receptors can work, where it's using the alpha chain to create the activity, okay, or to influence a nearby protein channel is using the alpha chain. That's one way, okay. And then it comes back over here and it has to be reset. And again, it takes energy to do a lot of the G protein mediated pathway kind of things. So that's again why you have a lot of mitochondria in your neurons to generate this energy needed for these conformational changes in proteins and so forth. So that's not the second messenger system. That's using the actual subcomponent of the G protein as a kind of a first messenger. That's like it does its thing just using an alpha subunit of the G protein. That's not the second messenger. Okay, now we'll talk about a second messenger system, which is a little bit different. In this case, the G protein, and I don't know whether it could be with that alpha subunit or whatever, we don't, or one of the other subunits. Whatever, it activates another intracellular target protein, not a membrane channel per se, but usually some type of an enzyme. Okay, that enzyme then produces another substance called the second messenger, which that second messenger then goes to do whatever activity within the cell that it's going to do, which could be open a membrane channel, ion channel, just like this alpha subunit did here. That's one thing it could do. However, that second messenger could actually go down into the nucleus, activate genes to say, hey, this neuron is going to be being used more often in this pathway. I want you to create more protein channels on the membrane of this neuron so that it's more excitable. Or it may do the opposite and say, let's get rid of some. They activate genes to say, let's cleave off and get rid of some protein channels because long term we don't want this neuron to be as active. So it can do more long-term regulation of a neuron by upregulating and actually causing your nucleus and gene activity to then change how many receptor sites and how many protein channels you have on that cell, either turn them up, turn them down, It could tell you to start producing more neurotransmitter in the synaptic vesicles that are going down there, so it becomes a stronger response, or it could say take more out. So these are more activating genes that causes increase or decrease. So I should say increases or decreases in synthesis of cellular products. Or it could regulate calcium concentrations in the cell. Calcium within your neurons, it's like stoking the fire. It's like, you know, coal in the furnace. The more calcium that's in your neurons, the higher the metabolic rate of your neuron is. So it's going to generally increase excitability or the activity, whatever that neuron does. If that's an inhibitory neuron, it increases its inhibitory nature. If it's an excitatory neuron, it increases the excitation of it. By just putting more calcium in the cell, it's like, you know, it increases the metabolism of the cell, stokes the fire of whatever that cell normally tends to do. It ramps up the processes in there. Okay. So let's pictures. I like pictures. Pictures always make things better. So here is a protein that's embedded in a cell membrane. Here's the G protein. In this previous picture, it showed the G protein intimately connected to that other membrane protein, okay, receptor protein. In this case, they show a gap. Don't worry about that. Just know that we could have this thing right over here, kind of bound almost to it, but we're showing some separation in here. And this is showing you kind of the, you know, 3 different parts: the alpha, gamma, beta parts of the G protein, just the 3 complex components there. And then this is another protein, and in most cases it's a protein that is an enzyme, okay? And you can see it's bound to the inside of this cell membrane. So let's do this. Let's draw a protein channel over here, okay, which has a gate in it, let's say. So we already mentioned how the G protein itself with the alpha component subunit, if this neurotransmitter binds here, it could cause this alpha subunit to float right on over here and directly activate and open that channel. Okay, open the channel. Okay, so that's one way. That's the first messenger. The first messenger is the actual subunit of the G protein. Okay, that's one way. Now we're going to talk about second messenger system. So in this case, it could be the alpha subunit, it could be the gamma. I don't know. It doesn't have to be the alpha subunit, but so this activates the G protein. Then the G protein, in some way or another, activates, instead of activating directly to the protein channel, it activates an enzyme nearby. That enzyme then produces whatever molecule now is called the second messenger. That second messenger, and we'll give you some examples, that's the arachidonic acid system and so forth, but it can then do a number of things. It can go up here and directly open protein channels, okay, and allow whatever, sodium, potassium, calcium, to come through. It can go to the nucleus of the cell and activate genes to then say, hey, let's put more protein channels, or let's make more neurotransmitter, whatever we need to do. That's another thing it can do. Or it could go in and actually help. You actually have internal calcium stores within your neurons too, so it could help release those internal calcium stores to just upregulate overall metabolism of the cell, okay, via the second messenger system. Is that helpful? Does that make sense? So the G protein is the first messenger, then it activates an enzyme which creates another molecule that's just second messenger, okay? So here's a nice little schematic that just shows the different. Here's by far the most common thing that happens in your nervous system. Receptors on ligand-gated channels, right? You have these receptors on ligand-gated channels. When a neurotransmitter or ligand binds to it, they briefly open, do whatever they're going to do, okay? Cause local depolarization or hyperpolarization, okay? G proteins, mediated receptors, either just the alpha chain itself via, or the second messenger, can cause prolonged opening of ion channels. So this is fast opening of ion channels. This is more prolonged opening of ion channels. Directly through the alpha chain or second messenger can do that. And that can also cause local depolarization or hyperpolarization, but for longer periods of time. Okay? The other thing the second messenger can do is it can go into the nucleus and activate genes that, again, can increase the rate of synthesis or decrease the rate of synthesis in specific cellular products. That will affect the overall activity and function of that neuron or cell. Or it can cause modulation of overall levels of calcium within inside the cell that just tends to upregulate or downregulate metabolism of the cell. Those are the different things that can happen via the G protein-mediated pathways. The G protein-mediated pathway is a little more nuanced. It gives you a little more, and it tends to affect more long-term activity in the cell, in the neuron or whatever. Where the first one is just rapid communication. That's just how we do the rapid, immediate, ongoing communication between neurons, muscles, and so forth is through that ligand-gated channels. Longer-term changes in neuroplasticity and overall function of regions of the nervous system and excitability that lasts minutes, hours, day, is G protein mediated. Okay. So here's just an example of one of the second messenger systems in our body, and there's probably hundreds. I don't know if there's thousands or not, but there's lots of these. One is called the cyclic adenosine monophosphate, or the cyclic AMP. I don't know if some people may have heard of cyclic AMP system. So in this case, you have activation of adenylyl cyclase, which is an enzyme in your cell that converts ATP to cyclic AMP. Okay. Cyclic AMP then modulates membrane receptors and gene expression. So cyclic AMP is the second messenger in this. So let's walk through it here. This is in the case of the cyclic AMP system is not just in the heart, but this is one example of how it works in the heart. Let's say you're exercising, your sympathetic nervous system turns on and says, "Hey, I got to increase heart rate." Okay, so you have nerves coming from your sympathetic nerves in your thoracolumbar area, right? Okay, and they exit out, go to the heart. And they're usually in that area of like T6 to T9 or so, but you've got some neurons, some axons that come out from there that are sympathetic axons that go to connect to your heart and supply your heart muscles, and it dumps some norepinephrine onto your heart muscle cells, okay? A neurotransmitter, norepinephrine. And it binds to G protein-mediated receptors on the heart muscle cell. Okay. Those G proteins then are activated, and when they're activated, they go over and tell adenylyl cyclase to ramp up its activity. Adenylyl cyclase is a enzyme which then creates and takes ATP and converts it to cyclic AMP. So we're increasing this production of cyclic AMP. Cyclic AMP is now our second messenger. It was created by that increased activity of the enzyme to create the cyclic AMP. Cyclic AMP, then that second messenger system opens calcium channels. And that opening of calcium channels causes your heart to contract, right? Increases heart muscle contraction because calcium is very important for contraction of heart cells and muscle cells, smooth muscle cells and heart cells and so forth. So that causes your heart to beat faster. This takes, though, a few seconds to a few minutes, right? When you start to work out, your heart rate doesn't instantly go up because there's a little ramp up. And then same thing when you cool down, even though you take away the signal, this modulating activity lasts for, it takes a few seconds to a few minutes for your heart rate to come back down, partly because you're using a second messenger system to do that. Because you don't want your heart, though, to either turn right on and right off like that's hard on it. So that's a way of kind of giving a little bit of a ramp up and a ramp down kind of system to kind of, so your blood pressure is not doing crazy things and ramping too high and falling off too fast. So this is just the second messenger system between a neuron and a heart muscle cell. Okay, one example is the cyclic AMP. And then here's another one that's more in the periphery here. These second messenger systems are not always associated specifically with neurons and muscle cells. These second messenger systems can occur in other areas or areas where activity within your modulating activity of cells. So, and this is the arachidonic system. This is where you get histamine binding to a G protein mediated receptor. Okay, so let's say you have, you poke your finger or you smash your finger or something like that, and you're creating inflammation. When you poke something in your finger with a knife or a pin or something like that, you're damaging cells, right? And cells tear open, and most of your cells have some histamine inside of it. As long as the histamine's inside of it, it doesn't do anything. But once the histamine gets released from the cell, it starts to circulate around to other cells nearby that aren't ripped open or damaged. It can activate them. This histamine's going to bind to some receptors on other cells that are nearby. And it's going to bind to a G protein mediated receptor, which that will cause, when you activate the G protein within that cell, it causes phospholipase A2, which is another enzyme, to create and make arachidonic acid. So now arachidonic acid is our second messenger. Arachidonic acid will actually go and increase production of what's called prostaglandins, and then that causes vasodilation. That's why you get swelling, or so this is binding to the cells on the walls of your vessels that cause them to cause vasodilation and relax some of those muscle cells, so it causes vasodilation. So this is how a histamine response can cause swelling and vasodilation and that red, feeling in your finger or whatever from the inflammation and damage. This is one element to that, okay? And this thing can happen like in your nasal passages and stuff like that when you get really stuffed nose and you use an antihistamine for an allergy or something like that. The G protein mediated pathway is involved with some allergic responses, and that can cause some of this vasodilation and so forth. So that's just another example. Aspirin and other NSAIDs. So this is why for pharmacology, they inhibit the enzyme, the phospholipase 2 enzyme in the G protein mediated cascade, which decreases prostaglandin formation. Okay, so that's what NSAIDs do. One of the primary actions is to impede or inhibit this second messenger system for causing inflammation and so forth. Good? Let's take a break. And then we'll be able to wrap. So neurotransmitters and also their receptors. Get into a little more specifics here. And this is where we're on kind of this bigger diagram on this page is kind of where we're at with talking about most of these, not all of them, but most of them. So neurotransmitters and neuromodulators. Most neurotransmitters / modulators are classified as either amino acids or their derivatives, amines and peptides. One exception is acetylcholine. It's a cholinergic neurotransmitter. Neuromodulators, while technically a neurotransmitter, tend to be longer acting and or cause changes in larger networks of neurons, and they utilize a G protein mediated pathway to cause their effects. Okay? So here's common neurotransmitters and neuromodulators. So neurotransmitters, fast acting most of the time. There's always the exception or two here, but for your purposes, just know that these typically are fast acting. Acetylcholine's unique because it can be classified as either a neurotransmitter or a neuromodulator. Acetylcholine in the peripheral nervous system tends to act like a neurotransmitter, and it's because it's fast acting at the neuromuscular junction, okay? And also in the autonomic nervous system, its role plays in the autonomic nervous system. GABA is one that's also a neurotransmitter, glycine and glutamate. If you know that all the things that start with G are basically a neurotransmitter, that's helpful. And just acetylcholine can be both, depending on whether you're CNS or PNS. So acetylcholine's the exception, but if it starts with a G, it's a neurotransmitter. If it starts with anything other than a G, other than acetylcholine, then it's a most likely, 90% of the time, it's going to be a neuromodulator or G protein mediated type neurotransmitter. So the neuromodulators, the slower acting ones, dopamine, serotonin, norepinephrine, substance P, endorphins and encalphins, and then acetylcholine as it plays its role in the central nervous system. It acts as a neuromodulator in the central nervous system. So when we talk about some of these neuromodulators affecting larger networks of neurons, and we're going to get when we get into the brainstem and the cerebrum, you're going to see some of these very similar pictures. You don't need to get caught up in the details here at this point, but understand that there are certain places in your central nervous system where you have certain regions that contain a fair amount of cell bodies that have axons that project to larger regions or networks of neurons within other regions of your central nervous system. So let's just talk serotonin right up here, up on the upper left there. You have a couple areas in your brainstem, okay, the raphe nuclei in particular, that actually have axons. So you have cell bodies in these raphe nuclei that are located within your brainstem. Those cells produce serotonin, and they have axons that project. And you'll notice that this has axons that project to very large regions of your nervous system, almost your entire cerebrum. And then the raphe nuclei also project down the lower part of the raphe nuclei, the inferior ones, project down to the spinal cord. You actually dump some serotonin into your spinal cord to control some of the activity and output of your spinal cord, and also part of what's called the descending pain control system to help modulate pain in the spinal cord through some serotonergic pathways. But anyway, those are you tend to have, you do have cells in other locations that secrete serotonin, but you tend to have a couple regions in your brainstem in particular. There's a lot of them grouped together there that project axons diffusely throughout your nervous system. And those raphe nuclei are squirting serotonin kind of all the time at basal rates into your nervous system. And as long as they're squirting the right amount into the regions that they're supposed to, then you have normal neurologic function, normal mood regulation, normal cognition, all these different things that this helps to modulate. But if it gets messed up, then you can have depression, you can have bipolar disorder, you can have schizophrenia, if some of those larger networks of neuron get impacted, okay? So that's serotonin. If we go over to norepinephrine, norepinephrine is also secreted by a couple areas that are in your brainstem. Your brainstem is just amazing. There's so much crap in that brainstem, and then we start cross-sectioning through that brainstem, and you see all where the cranial nerve nuclei are in there, and then you realize there's all these other areas or regions in the brainstem that are important for secreting some of these larger, networks of neurons, of neurotransmitter. But anyway, you have a couple other things. This is called the medial reticular zone and the locus coeruleus that are down in the brainstem. They have axons that, again, project to very diffuse regions, similar to the serotonin system, throughout the cerebrum and even down into the spinal cord area and even to the cerebellum, okay? So norepinephrine is secreted throughout the central nervous system, with a preponderance of it coming from a couple of these localized regions of groups of cells in the brainstem that are really important for secreting that basal amount of norepinephrine, okay? We also have acetylcholine in the central nervous system here. This is just acetylcholine in the central nervous system. There's a nucleus called the pedunculopontine nucleus here that plays a role in secretion of acetylcholine into a little more selective regions, kind of through this medial forebrain bundle, into this inferior cortex here, frontal cortex area, and also into the hippocampus for helping with memory, but then also down to the brainstem and even into the cerebellum, okay? Acetylcholine. And then we have dopamine. Dopamine is coming from a select region in the midbrain called the ventral tegmental area. A lot of that goes to the prefrontal cortex. Some of that goes to the basal ganglia. When we talk about Parkinson's disease, dopamine is important for regulation of movement as it's getting kind of squirted into the basal ganglia circuitry. As long as you have a certain amount of dopamine being constantly squirted into your basal ganglia circuitry, it maintains your certain amount of motor output overall. We'll talk about that. But also, dopamine's important. It goes to this prefrontal cortex through the medial forebrain bundle. It also supplies amygdala, but this is, again, pleasure, reward centers, and so forth. So dopamine's important for that. Okay. So some of our drugs that mimic that or antagonize that are the ones that are most abused because it's going to our reward centers, makes us feel good, get dopamine squirted into those areas. So we'll just talk about some of these more common neurotransmitters and neuromodulators. The first one we'll mention is acetylcholine. It plays a major role in transmitting information in the peripheral nervous system, including at that neuromuscular junction. Okay, that's what helps you contract your muscles. Also in the autonomic nervous system, acetylcholine plays an essential role in autonomic nervous system transmission, both in the sympathetic and parasympathetic systems, and we'll get there a couple of weeks from now. We'll talk about the role acetylcholine plays there. And that tends to be mostly fast-acting, okay? So in the peripheral nervous system, at the neuromuscular junction, or within the autonomic nervous system for connection between some of the preganglionic, postganglionic neurons and all that stuff, it's mostly fast-acting acetylcholine, okay? In the CNS, which we showed you in that last slide, where it kind of influences those little bit larger networks of neurons, it's more neuromodulating, more longer-acting neuromodulating features. GABA. So it starts with a G. It's a neurotransmitter. It's fast-acting. It happens to be, it stands for GABA aminobutyric acid. It is the major #1 inhibitory neurotransmitter in the central nervous system. It's the fast-acting inhibitory neurotransmitter in your nervous system. Okay. It is an amino acid. There are two primary receptors it can bind to, GABA A or GABA B. You tend to have more GABA A receptors or more in your cerebrum. GABA B receptors tend to be more in your brainstem and spinal cord. Okay, so there's different areas of your nervous system that have slightly different types of receptors for GABA. They're still inhibitory, but just that, you know, so the neurotransmitter itself can influence activity, but then the type of receptor it falls in can influence the activity that occurs in the next cell. So GABA A, when you have GABA binding to a GABA A receptor in your brain most of the time, in your cerebrum, it causes chloride channels to open, causing hyperpolarization. That's how it causes inhibition. It creates the neuron, makes the neuron hyperpolarized. Benzodiazepines, things like Valium and also barbiturates, they are GABA A agonists. And so things like Valium, like if we give our patient with, like a lot of my patients have a lot of tone or spasticity. If you give them Valium to try to decrease spasticity, it actually works really well for. Problem is it binds to a lot of the GABA A receptors. It makes you really fatigued and out of it. You know, you give Valium as a depressant, central nervous system depressant, even though it works kind of like making your muscles less spastic, it makes you so out of it and fatigued, it's hard to get good therapy done, and it makes, you know, it just doesn't, it's not great for that. GABA B, this is linked to ion channels via, this is a little different. This is why I say mostly. So GABA B, which is mainly more in the brainstem and spinal cord, it uses second messenger system. You know, this is where GABA is typically a fast-acting neurotransmitter most of the time, except when it falls on GABA B receptors, it actually does use a G protein-mediated pathway. There's a medicine called baclofen that you can take either orally, or they actually put a pump in your spine and it squirts onto your spinal cord. Or if you take it orally, it will mainly bind to GABA B receptors. Baclofen is specific. It's an agonist to GABA B receptors, so it downregulates CNS activity at the spinal cord level, has less effect on the cerebrum. So that's why they use baclofen a lot to decrease people that have a lot of spasticity and tone from like a stroke or not Parkinson's, but stroke, brain injury, sometimes CP, cerebral palsy. They will give baclofen or a baclofen pump to bind to the GABA B receptors and cause their muscles to weaken. It does weaken your muscles, but it makes them less spastic, so you can hopefully move them a little easier despite them being a little weaker, but it doesn't cause the profound effects on your cerebrum because it's not binding as much to GABA A's. It does a little bit, so you still feel a little fatigue, and some people, there's a little transition period when you're first starting taking baclofen where you feel some more effects of fatigue, but over time it's less of an effect, but it doesn't have that profound effect like a Valium or something like that does. Glycine starts with a G, so it's a neurotransmitter, fast-acting. It's also amino acid. It inhibits the postsynaptic membranes primarily in the brainstem and spinal cord. That's all you got to know about that. It just causes inhibition of the postsynaptic membranes. Most likely, I think it's from opening chloride channels, so a little bit more negative, charge comes in, so it hyperpolarizes. So, glutamate. So this is the opposite of your GABA. So underline this. Most major excitatory neurotransmitter. So it's the counteractant. This is the opposition to glutamate and GABA are your two big, GABA's the major inhibitory neurotransmitter. Glutamate's the major excitatory neurotransmitter. It's also an amino acid. It activates the N-methyl D-aspartate, or NMDA receptor on a cell. NMDA receptors are also implicated for neuroplasticity in this thing we call long-term potentiation, where you get long-term changes in the receptor activity so that you enhance activity between cells or connections between cells. Glutamate is also implicated in what we call excitotoxicity and death of neurons with overexcitation. So let's say you have a really big brain injury, and let's say it's a car accident, and you'd go through this deceleration kind of injury where your brain gets pushed forward, your frontal poles of your frontal lobe and your temporal lobes whack into the front of your skull, plus you get whipped backwards so the back of your brain gets bounced around, plus you shear axons. The axons that are going down, the big white matter axons going down, they get sheared off. What happens is you shear all these axons, you damage these cell bodies, they just dump all their neurotransmitter into the region around where the damage is. And they have glutamate in them. So a lot of your neurons have this glutamate. So excess glutamate just gets dumped in to neurons all around the area of injury, and that causes overexcitation, could lead to things like seizure activity and so forth. But you're dumping a bunch of excitatory neurotransmitter into cells that are nearby that may have also got kind of shocked in this trauma, and they kind of go into a little bit of a haywire kind of a, form of shock, and they don't do a very good job of dealing with all that glutamate. You're trying to stimulate an injured cell that is nearby where these cells have been ripped open, and then you can actually cause those cells to die because they have to be more active than they're ready to be, and you actually can cause them to die off by killing them off with too much excitation. And so some medicines they use, or when they do like core temperature cooling, or they use some of the steroids in a patient after a brain injury, is trying to get rid of the glutamate, try to break the glutamate down so it doesn't cause the overexcitation, sequelae of the trauma. Dopamine. It definitely has an effect on motor activity and the role it plays in the basal ganglia, which we'll talk about. But it also plays a role in motivation, cognition. Our pleasure reward centers are, influenced by dopamine heavily. It's a monoamine. The major source of dopamine is that area within the midbrain called the ventral tegmental area. In the midbrain, all the way through your brainstem, kind of the meat of your brainstem, in your midbrain, pons, and medulla, there's like this area in the middle called the tegmentum. This just happens to be in the very front part of the tegmentum in the midbrain. That's why I call it the ventral tegmental area. It's just a region kind of in the middle of your midbrain, but slightly more forward in this area called the tegmentum. And that's where there's a more specialized structure we call the substantia nigra that's in there. That's the one that supplies the dopamine to the basal ganglia circuitry. That's part of the ventral tegmental area. There's different regions of the ventral tegmental area that supply it to that through the medial forebrain bundle up to that prefrontal cortex and those reward centers. Loss of dopamine and actually dying off of the cells in that ventral tegmental area in that substantia nigra, and those cell bodies die. you lose the dopamine that's being squirted into the basal ganglia or released into the basal ganglia. Serotonin. Serotonin adjusts the general arousal level and suppresses sensory information. For example, serotonin plays a role as part of the descending pain control pathway, so it actually inhibits pain and sensory information coming in. Low levels of serotonin are associated with depression, okay, and suicidal behavior. It's a monoamine as well. Serotonin is a monoamine. Neuromodulator. This is G protein mediated pathways that this is acting on. Longer acting, influencing larger networks of neurons. Norepinephrine. Norepinephrine is critical and plays a vital role during active surveillance of surroundings. When norepinephrine is being secreted at its highest levels, and this fluctuates during the day and also whether you're awake or not versus when you're asleep, obviously, you have higher levels when you're awake because you need to be actively surveilling and vigilant. The lowest levels are during sleep. Norepinephrine binds to alpha and beta receptors. So there's alpha and beta receptors. Norepinephrine, we know, also plays a role in the autonomic nervous system, in the periphery. Okay, we'll talk about norepinephrine and the role that plays in the autonomic nervous system, and particularly the sympathetic nervous system. Most of your sympathetic nerves release norepinephrine on their effector organs, whether it be a muscle cell or gland or something like that. The norepinephrine, and that typically is still a G protein mediated pathway, even in the periphery. Like we gave the example of the arachidonic acid pathway for the heart. That's a norepinephrine mediated, but it's still a G protein. It's in the periphery. Versus acetylcholine tends to act more like a neurotransmitter in the periphery. Fast acting norepinephrine, whether it's acting in the central or peripheral nervous system, tends to be more G protein mediated pathways. Substance P. Substance P happens to be a significant concentration within the spinal cord, where it acts as a neurotransmitter in the nociceptive pathway. However, at other sites, substance P acts as a neuromodulator. Okay, so it's only in the spinal cord that it really acts like a neurotransmitter. But everywhere else in your central nervous system and the cerebrum and so forth, it acts as more of a neuromodulator, usually producing long-duration excitation of postsynaptic membranes. So it's an excitatory neurotransmitter, and it's a peptide. Substance P peptide. Endorphins. Endorphins are found in areas with opiate receptors, including the substantia gelatinosa, hypothalamus, periventricular gray. You don't have to know any of this stuff right now. Periaqueductal gray. These are just areas within the central nervous system. Some of that you recognize, some you don't. The primary action is inhibition of slow nociceptive information. So what happens is, in one of our pain responses, if you have pain and the pain lasts more than a few seconds to a few minutes, eventually, if your body's working right, your body will naturally start to secrete or release some endorphins, which will bind to regions within your nervous system to inhibit pain. Okay, so endorphins, and also there's also called encalphins. And I think those encalphins are on here. I have them listed here. And I wrote in whether they tend to be, whether these neurotransmitters tend to be inhibitory or excitatory, some or both, depending on the type of receptor it falls on. Encalphins in this pathway, you could just draw a line up there. These all, endorphins and encalphins all fall on, we're just going to collectively call them opiate receptors. There's all kinds of different types of opiate receptors, but we're just going to call them opiate receptors. So this is a table from, I don't know which edition this is from, and this, I think this is probably from the 3rd edition. And I don't know if it's changed in the more recent edition or if they've gotten rid of this table in the 5th. I forgot to check on that. But so you have your neurotransmitters, okay, or neuromodulators. So they're calling everything a neurotransmitter here, but we know that it's really mainly just these guys that are primarily called neurotransmitters. The rest of these are more the neuromodulators. But again, they can all be classified as neurotransmitters. But then they have receptors they fall on. you know, most have more than one type of receptor, which can influence the action of that. Okay, in some cases, it actually can cause the neurotransmitter to be inhibitory versus excitatory, depending on the receptor it falls on. And when we get to the basal ganglia circuitry, when dopamine falls on D1 receptors, is excitatory, but when it falls on D2 receptors, it's inhibitory. Okay, so just understand that it's really complex, really nuanced how all this stuff works together. For all we're doing is turning things up, turning things down, turning certain regions up, turning certain regions down. It just gives us lots of ways to do that in a very nuanced way. It's very complex. Some clinical considerations before we wrap up here. When you guys are in pharmacology, when you get to this section, when you start talking about medications that are agonist or antagonistic to neurotransmitters. So a lot of medications out there are trying to mimic, you know, or block one of your naturally occurring neurotransmitters, okay, to increase or decrease effects of that. So examples. Botulinum toxin A, it blocks the release of acetylcholine. So it's kind of acts as an agonist, antagonistically to acetylcholine. It's not really classically an antagonist, but it blocks the release of acetylcholine into the synaptic cleft, the neuromuscular junction. So when you get Botox, make sure it weakens your muscle. Okay, so that's kind of how it makes your facial features. If you get Botox in your face, it makes your muscles relax, which makes your wrinkles look smoother or less prominent when you do that. If you do it to somebody who's got a lot of spasticity or tone, most of the people I see with Botox is they do it in their calf muscle because they have a lot of tone in their calf muscle that impacts their walking, and they get this thing called clonus or spasticity where their leg jumps around. So they might inject botulinum toxin into the calf muscle to make it less jumpy, decrease the tone, but it weakens it too. So they can't use it voluntarily as much, but it's now not jumping around on them as much. So there's always trade-off with this kind of stuff. Okay. So it's blocking the release of acetylcholine. Baclofen, it's an agonist for GABA B receptors. So remember the baclofen, the GABA B receptors tend to be in the spinal cord region and some of the brainstem. So you're taking it either orally or you thread this little catheter so it only will squirt right on the spinal cord. If you take it orally, it gets everywhere through your bloodstream, including into your cerebrum, where it can cause a little bit more of that, effects of making you feel a little loopy or tired or fatigued. If you squirt it right onto the spinal cord, it doesn't get up to the brain, and it has its actions just at the spinal cord level. And it binds to those GABA B receptors. It's a molecule that's very similar, so it wants to bind to those GABA B receptors and cause inhibition. Propranolol is a beta blocker, right? It's a blood pressure medication. All those the ones with OL on the end tend to be beta blockers. They're antagonists to beta 1 receptors. They block or they antagonize the beta 1 receptors because they don't let them bind, and they block them. So they block the receptors for norepinephrine. So norepinephrine can't bind to those, which norepinephrine is part of that sympathetic nervous system, which increases heart rate. It actually causes some vasoconstriction, we'll talk about. So it's basically decreasing heart rate by binding and blocking those norepinephrine receptors, so you can't increase your heart rate. It limits it a little bit, modulates a little bit, and also causes some vasodilation as well. So you get lowering of blood pressure, lowering of heart rate, because you're blocking beta 1 receptors from the stimulus of the sympathetic nervous system through the beta 1 receptors. Synaptic is a medication that is it's a medication that's a form called L-DOPA that once it's in the central nervous system, it converts to dopamine. It's a precursor to dopamine. You can't just give somebody dopamine actually like as a medication because it doesn't cross the blood-brain barrier. L-DOPA is a small enough molecule, it can cross the blood-brain barrier, and then it gets converted by tyrosine into dopamine within the brain itself to have its effect. Okay? So that's Synaptic. So it's a dopamine, acts like a dopamine agonist. Eventually it's turned into dopamine, so it actually truly becomes dopamine eventually, but it's functioning essentially like a dopamine agonist, trying to increase the effect of dopamine. Different classes of medications that you guys have maybe seen and you'll hear about in pharmacology. SSRIs, also known as selective serotonin reuptake inhibitors. So they're basically inhibiting the reuptake of serotonin. Once serotonin's dumped into the synaptic cleft, it keeps it there longer. So it's more likely to bind to all the available receptor sites and stay there a little longer, versus being taken right back up into endocytosis or having the glial cells take it. It helps to maintain and keep the enzymes from breaking it down and keeping it in that synaptic cleft longer. So whatever serotonin you have, it stays. You enhance its effect and maintain its effect a little longer. So if you have depression and they think your serotonin levels are a little low, this is one way to compensate for that by keeping the serotonin you do have in that synaptic cleft longer so it can do its job better. Does that make sense? So that's a selective serotonin reuptake inhibitor. Also, there's another class that does a very similar thing, is MAOIs, monoamine oxidase inhibitors. Those also prevent breakdown of serotonin, dopamine, and norepinephrine. All those, because all of those are monoamines. Okay, so it helps all those to stay in the synaptic cleft and bind more effectively in that area. So, and it's definitely an art and sort of a science with trying to balance those medications out. Anybody that's gone through that process, it's can be very frustrating, and just when you think it's working, it can sometimes then not work, and then they have to alter it and try different medications, trying to get that, because it's not often just one neurotransmitter, it's all three of those, and then certain medications will change the levels of those differently, and that in an individual person, it all works differently because your genetics and how your system responds to those medications. And it's often a trial and error process, and the problem is you have to wait 2 to 3 weeks to get the effect, and by then, now you go, okay, this is not working, and then you have to wean yourself off of it to try the next one, so this takes months, and so it can be a very frustrating process for people with trying to deal with some of that. Yeah?
So let's say you're taking an SSRI and cut cold turkey, like why do you even wean yourself off?
I would think, so it's prolonging the life of that, but so your receptors then adjust their level of sensitivity based on how much is in that, how much is now staying in the synaptic cleft. And so if you get your receptors, if you keep more in the cleft, the receptor will then eventually get used to that amount in that, and it'll adjust the sensitivity to how strongly it'll bind. But then if you now take it away and now your receptor sensitivity is not a match for how much, and so it can just alter how, I don't know the exact ways, but that's, what I can see is happening is that now you have less staying in the cleft, but that you have these receptors that are also now less that have less affinity, because they actually you'll downregulate. If you got more staying in there, they won't quite as they won't have quite as much sensitivity or affinity. And then once you suddenly remove it, and now you have these receptors that now don't have as much affinity for even a lower level now in this cleft, and that can mess you up. So it takes a while then for that all to get back to baseline again. So diseases involving synaptic transmission and neurotransmitters, all kinds of them. Here's a few examples. Parkinson's, we mentioned, with dopamine, lack of dopamine into the basal ganglia circuitry. Lambert-Eaton syndrome, one cause of it can be small cell carcinoma of the lung. Actually what happens is your body, in its immune response to try to attack the cancer, it'll create antibodies to calcium channels. So you'll feel weakness in your muscles because your muscles, calcium is important for muscle contraction, and so you'll feel weakness because you damage the calcium channels in your muscles because you've got these circulating autoimmune antibodies to calcium. It just mistakes your calcium channels. It's trying to create an immune response against the cancer, and it just happens that it tends to also affect the calcium channels. And it's very specific to the small cell carcinoma of the lung. That's sometimes how they find this cancer, is the person all of a sudden starts having weakness of their muscles and wasting, and they think it might be something like myasthenia gravis is more common, and then they check them out and they actually have small cell carcinoma of the lung. You can get Lambert-Eaton syndrome without cancer as well. Some people can have a Lambert-Eaton syndrome that just develop these immune responses and antibodies to calcium channels. But a common reason is that small cell carcinoma of the lung. Similar to kind of Lambert-Eaton syndrome, but it's not affecting the calcium channels in the muscles themselves, is the myasthenia gravis has antibodies to the acetylcholine receptors, okay, on the muscle cells. So it is on the muscle itself, but it damages the acetylcholine receptors. So when your nerve releases acetylcholine, there's just not as many acetylcholine receptors for it to bind to. So there's less chance you're going to get a contraction of that muscle. So it's going to, again, cause weakness of muscles. Okay. Especially the muscles that you have to turn on all the time. So it actually, like eye muscles, facial muscles, tongue muscles, those get the weakness first because those muscles are active almost all the time, and you'll notice, and they're smaller muscles, okay? So they're more disproportionately affected by changes in the number of, acetylcholine receptors in these small muscles. You start losing a few of those acetylcholine receptors, you really notice it right away, plus they're muscles that are active all the time. And also like proximal muscles, like muscles of the proximal limb girdle and spine, they're turned on all the time as well, so they're always craving acetylcholine to keep them contracted, and now you start losing some acetylcholine receptors. It's harder to keep them kind of recirculated and keep contracting those muscles. So myasthenia gravis. And then depression is a big one. Any abnormality in the levels of serotonin, dopamine, or norepinephrine can lead to changes in mood, personality, depression, those kind of things, and trying to maintain a balance between all those. So chronic pain can lead to changes in some of those as well. So if you're not sleeping regularly, let's say somebody has a lot of pain, which affects their sleep. Sleep is important for replenishing supplies of some of these neurotransmitters. If you're not sleeping a lot, your serotonin levels tend to go down, which serotonin then causes you to feel depressed. But serotonin does play a role in the descending pain control pathway. So now you have pain that you're less able to body naturally combat because your serotonin levels, plus you're depressed, and you get this vicious cycle, kind of thing going on with some of those things. Woohoo! Any questions on that stuff? It's not too bad. Just give it another read through. Tomorrow